Hello friends, good evening. Welcome to Sam Biology class. Friends, today we shall discuss about vernalization. While discussing photoperiodism, we came to know that numerous plant species can be induced to flower by providing them a suitable photoperiod. Similarly, flowering can also be promoted by cold treatment. Vernalization is the process whereby flowering is promoted by cold treatment given to a fully hydrated seed or to a growing plant. Dry seeds do not respond to cold treatment. Without a cold treatment, plants that require vernalization show delayed flowering or remain vegetative. Many of these plant species grow as rosettes with no elongation of the stem. The term vernalization was coined by a Russian geneticist T.D. Lysenko in 1920. This term is derived from a Latin word verne that means spring-like. Vernalization is also known as Yerovization or Lysenkoism. The process of vernalization results in competence to flower at the shoot apical meristem. The effective temperature range for vernalization is from below freezing point to about 10 degrees centigrade with a broad optimum from 1 degree centigrade to 7 degrees centigrade. Few plant species can also be vernalized at a low temperature from 0 degree centigrade to as low as -6 degree centigrade. Plant species vary considerably in their age at which they respond to cold treatment. Winter annuals such as winter cereals respond very early in their life cycle, whereas biennials and many other plants must reach a minimal size before they respond to low temperature. Site of vernalization. The perception of cold stimulus is thought to take place in the shoot apical meristem. Embryos or even isolated meristems from rye seeds have also been vernalized. When a vernalized meristem is transplanted on a non-vernalized plant, then it will ultimately flower. But if a non-vernalized meristem is grafted on a vernalized plant after the removal of vernalized meristem, then the plant will not flower. Next, devernalization. Vernalization can be lost if followed by certain conditions such as high temperature. This loss of vernalization is known as devernalization. The effective temperature range for devernalization is from 25 to 40 degrees centigrade. In case of winter rye, it is about 30 degrees centigrade. In few cases, revernalization can also occur if followed by another exposure to low temperature. Mechanism of vernalization. Two different theories have been proposed to explain the mechanism of vernalization. They are phasic development theory and hormonal theory. The phasic development theory was proposed by T.D. Lysenko in 1934. According to this theory, flowering involved two different phases, thermophase and photophase. Thermophase depend on a suitable temperature. Vernalization accelerates this phase. And the second phase, that is photophase, depend on a suitable photoperiod. So according to this theory, a suitable temperature and a suitable photoperiod are responsible for flowering. The second theory of vernalization is hormonal theory, which was proposed by Meltzer in 1939. According to this theory, in response to cold treatment, plants synthesize a hormone-like substance termed as vernalin, which induces floral transition. It is because of vernalin, grafting of a vernalized meristem with a non-vernalized plant also induces flowering. The physical and chemical nature of vernalin is not known. It has neither been isolated or identified. One more important point is that in many plants, gibberellin substitutes the cold requirement in induction of flowering. But it is clear that gibberellin is not vernalin. Evidences indicate that vernalization may involve epigenetic change in gene expression. Arabidopsis require both long days and vernalization in order to flower. In this plant, there is a gene termed as flowering locus C or FLC, which is a repressor of flowering. In non-vernalized shoot-apical meristem of Arabidopsis, this gene is highly expressed, as a result of which flowering is inhibited. But when a cold treatment is given, this gene is switched off, as a result of which the plant responds to long days in order to flower. And this genetic change, which remains stable even after the signal that induced it is removed, is known as epigenetic regulation. It is because of this stable change in gene expression the plant remains vernalized throughout its life cycle. So friends, this is all about vernalization. For more updates, please subscribe my YouTube channel, Sam Biology Class. And also please share your comments. Thanks for watching.